Hi, everyone. It's Nate O'Connor here. I'm a product manager for NeuroInfo with MBF Bioscience, and we're going to be running through a webinar today on registering whole mouse brain volumes from serial sections to the Allen CCF in our NeuroInfo program. I'm thrilled to be joined today by renowned neuroanatomist Dr. Chip Gerfen, who's on the line with us, and our own MBF computational neuroscientist, Dr. Brian Eastwood. So we'll be discussing kind of interactively as we go through. I do want to remind people that this is one in a series of webinars about this topic of mapping brain volumes into an atlas reference space. And our last couple were really general overviews, and they're available on our website, the MBF website, under resources. I'll give an address at the end too. But this one, we're going to run through a certain part of the workflow. So we're going to do a deep dive here. And so I just wanted to remind folks of the overview of the workflow, because there's some advantages to it that are worth noting. So we start with slides, we go to the scope, we image them somehow, and we end up with whole slide images that have our serial sections in them. We take and then compile those into a brain, we register and compile them into a brain volume that we then register into the Allen CCF and its atlas. And that's the area we're going to focus on today, is the highlighted blue square that I just put up there. And in this example, once I've registered my images to the Allen CCF, any measures I make, and here, if you look, their actual cell coordinates in the experimental space, I can apply the transforms that match the images, that register the images into the Allen CCF to the coordinates of the detected cell markers. And so then I can start to accumulate measures across animals in a common coordinate system. And Chip's going to kick off his part of things with a really cool case study that shows an endpoint and why, just the power you can have out of this particular technique. So really, to really drive home what the process is, we start here today. We're going to start when we already have that aligned brain volume from serial sections and compiled. And that's out of BrainMaker, which is inside of NeuroInfo. And what we do is we take a representative section from that brain volume, that has a lot of landmarks that we can use to semi-automatically find a good fit equivalent section in the Allen average mouse brain, and that's part 2 here. And so we're going to be focusing on part 2 and 3, and then once we have a relatively satisfactory fit, we apply that as a starting point for estimating transforms that register all of our rostral-to-caudal sections throughout the brain volume that we're working with. And Chip's going to go into a lot of detail for that. So there's a couple things I just want to point out that were really simple things that we discovered early on in the research of this. And all of this is developed from NIH-funded research, and I give those original grants at the end, just if anyone's interested. Tissues aren't frozen or cut in the canonical orientation, so there's always an angular offset, and there's always distortion in the section that shifts us or scales us more appropriately from the imaging sampling space compared to the Allen sampling space. So this is a demonstration of that. So here's an experimental section in the upper left, and you can see I have equivalent sections from the Allen Atlas average mouse brain in the top right and the lower left. And if you look, you can see a couple things that are striking right off the bat. There's some compression in X, perhaps due to processing, but there's really marked compression in the axis that we cut along from the dorsal to ventral surface. So there's really knife compression happening. And we found in order to get a reasonable estimate and a reasonable amount of time of the registration to the Allen Atlas, we had to really compensate for that first. And so what we did was, for several brains in a given laboratory, this graph represents for 10 of those brains that the scale factor, that the adjustment to the scale factor along Y really had no statistical significant difference between brains in that protocol. What the take home from that was pretty impactful, because what it means is a lab can install NeuroInfo, take a representative brain from the protocol, calibrate it for that scaling between the experimental space and the atlas space, and then use that for every brain. So it's a one-time calibration of the system that you use, and then you can use per laboratory. Not surprisingly, if you go to other laboratories with slightly different protocols, you get different correction factors, but they are all, we're finding that they're all pretty consistent within a laboratory. So that was an impactful thing, and really impacted the direction that NeuroInfo moved forward in, is that you can start with the starting point of the scaling factor for every brain you go in, you work with. The other thing that we found that we had to compensate for, and we were able to, is asymmetry. So you're not, as I said, you're not freezing or cutting in a canonical orientation like the atlas is drawn in. Your sections are probably really between the pages of the atlas. And so I've got here on top an experimental section that shows some left-right asymmetry. And I've got on the bottom the discovered or estimated equivalent section from the Allen average mouse brain. And just as a note here, that red line represents in the sagittal and transverse views the angular offsets that we had to estimate to accommodate for the asymmetry in the experimental space, whereas we'd be between pages and trying to visualize that in our mind if we were in a paper or even static digital atlas. So just a couple of examples. We've been able to handle some fairly extreme cases of angular offset, and then Chip's going to jump in and in some of his presentation and talk about when linear methods aren't enough, or don't quite get you there with the fit. You can often employ a nonlinear mat registration to the Allen space and accommodate for things like physical deformation, like due to an injection depression or something like that here. So we're going to give examples of all of that. And Chip, with that, should be good to go. So everyone here is Dr. Chip Gerfen.
Hi, I'm Chip Gerfen. I just want to start off with showing an example of a brain that from an experiment. This is a pretty cool experiment that was done by Anna Lorenko Ignacio and Suyin Lee's lab in IMH. And what Anna did was that she's injected 15 or 20 different animals, different mice, with a single rabies injection into one neuron in the somatosensory cortex. And what we've done here is overlay the results from those 15 all registered to the Allen space. And this just gives an example of why the power of doing whole brain registration to a common coordinate framework. So this is a top-down view looking at the injections into the somatosensory cortex and of the 15. And then we can view them in 3D, and you can see they're all, the injection sites were all in the somatosensory area here. Then we can view them in the thalamus, the regions in the thalamus, in the cingulate and prefrontal cortex, contralateral cortex, and regions like that. So the real power of the ability to register to the Allen space is that we can combine and we can look at different cases in the same space. And so I'm going to turn off, and with this program we can actually turn off different different cases. And so this is a single case, and so this is the primary injection in the neurons. These are individual neurons that are projecting to an individual neuron in the somatosensory cortex. So all of these are transsynaptically labeled from this one injection neuron. And so this is most of the neurons that project to that are local in the adjoining somatosensory, primary somatosensory, but then there's a region in the secondary cortex and then the thalamus and prefrontal areas, contralateral areas. And then we can combine, look at other cases and see how they relate. Let me see if I can find one. Yep. So this is a second case, and we can combine that to see how the neurons are labeled in the different regions and the relationship. looking at things like the topography and the maintenance of topography and the areas that project into the somatosensory cortex. This just highlights, or meant to highlight, the real power of the ability to view different cases at the same time in the same space. And so a lot of quantification, things like that can be done. So with that, I'm going to switch to how we generate maps like this. And now I'm going to turn to a case that we've, this is experiments that have been done with Ali Girasol, Mike Ryan, in Alexandra Nelson's lab at the University of California in San Francisco. This is an injection of rabies starter, AAV rabies starter into the striatum in a D1 Cree mouse. So, and then the red neurons are neurons that are transsynaptically labeled that project to the D1 neurons in the striatum. And so this is a case where what we wanted to do was to look at the inputs from cortical areas and other areas into the, into D1 neurons in the striatum and compare them between different conditions. This is in Parkinson's model. So compare the projections from the cortex and thalamus and globus pallidus to the D1 neurons in normal animals, animals that have been treated with 6-hydroxydopamine to deplete dopamine in Parkinson's models, and then animals that had been treated with L-DOPA that display dyskinesia. So the power, what we wanted to do was to be able to then register or count the neurons in different areas of the brain from these different experimental conditions. So this is a single section through a stack of, from one of these brains. This is a control brain, actually. You can see the injection site of the rabies. And I just want to go through the windows that are in the program. So this is the NeuroInfo program. The main image is shown in the main window. Then there's this orthogonal view, allows us to move between different parts of the whole image stack. Then there's this image organizer, tells me what case this is that I'm looking at. The image adjustment window allows us to turn on and off the different fluorophores. So there's all of the brains that we use have been stained with a fluorescent blue Nissl stain. So we can turn that on and off. Turn off the rabies labeling and just see the starter neurons in the striatum, see this injection. And then we can also move around in the image. And in this box, I can go to the injection site, go up to the cortical areas where these are neurons that are labeled.
So Chip, can I ask a question? I see that you're able to clearly resolve here individual neurons. What were these scanned at? What magnification?
So most of the experiments that we do are done with a 10x objective.
Okay.
We find that that's typically sufficient to be able to detect neurons. So in this program, we can also detect the individual neurons. And I've done this before. We're going to have a webinar on this separately to show that, but we can see these, can see the neurons. So see, there's a pyramidal neurons in the cortex projecting to the striatum. And these are the cells that we're interested in seeing and registering to the Allen space so that we can measure, count the number of neurons in every area and compare between different, let me just change the size so they might show up a little bit better. You can see those, okay? So anyway, so this automatic cell detection method enables us to view the cells. So over here, I can change the size of the markers. I'm going to view, then pan out a little bit. So in this window, we can navigate and change the markers and either view markers or turn them on and off. And in the actual experimental brain case, brain images. And so, as I said, we can go to the front of the brain, looking where the cortical neurons are there. and I can actually, scan through. So this is, this whole brain volume was generated by taking every coronal section through this brain and then aligning them, registering them to each other to create this whole brain volume. We call it a stack. It's an image stack that has every section through the brain, and it creates this 3D volume, and then we can scan through it. So we're going caudally. You can see neurons in the globus pallidus here, neurons in the cortex. Come back and start to see neurons in the thalamus, interlaminar thalamic nuclei, and even back in the parafascicular nucleus. So this is the 3D volume that we've generated from these serial sections. In this case, the distance between the sections is 50 microns. So typical sectioning, distance that we use. The other thing we wanted to do was to put fiducial markers into this brain. And so these are yellow. So these are markers that I laid down to, on fiber tracks, the corpus callosum, the outside of the cortex, the anterior commissure, things like this. So I use these fiducial markers that after we do the registration, we can see how accurate the registration of these sections is to the Allen space.
So it's not actually using them to do the registration. They're there to see, once they're transformed into the Allen space, how accurate of a job we did.
Exactly. So these are, markers that are placed around the globus pallidus. We can see the neurons in there. So I'm going to turn off these markers. Go back up to an area just to show the neurons that have been marked. So these are all the, so again, what we're going to do is to register that atlas volume or the image volumes to the Allen space. So now I want to go through that process. So in this window, this allows us to navigate through the experimental images themselves. Make them larger, smaller, and all that. The next part of the program, or the program that does the registration, is under a toolbar called section registration. The whole brain volume is generated with the Brain Maker workflow, and as I said, we're going to go through a separate webinar and go through the details of that. But for the purposes of this, I want to show how we do the section registration to the Allen space. So when we do that, it opens up new windows. These windows allow navigation through the Allen coordinate space. One thing I should say is that what we found is that when we register to the Allen volume, the original Allen space, that space was generated from at the Allen Institute from over 1,000 brains that were averaged together. There were 2 photon images. There were block face images, basically coronal block face images through the whole brain. And that was what was used to generate the coordinate system of the Allen Common Coordinate Framework Atlas. What we found is when we do the registration, almost all of our brains are Nissl stained. And so we actually, the images are different than the 2 photon images. And so we generated an average Nissl brain that was then very precisely registered to the Allen space. So the coordinates in our Nissl brain, our average Nissl brain, which was generated from 70 to 80 average, or 70 to 80 original brains, that's what we use to register. The program does enable using different starting atlases for registration. The one we're using now is an average Nissl. So this is a window.
If I could just jump in there, Chip, just to say, we also do support cross-modality registration. So if your modality that you're using doesn't exactly match the reference image in the atlas, we can still do the registration. But in this case, because we have a modality that matches what you use in your lab, it's a good reference to use for doing the registration.
Yeah, so we could, what Brian's saying is that we could actually do this registration against the original 2 photon images. And for the most part, it works pretty well. We just find that we get a little bit better results when we use the modality that matches what our experimental sections are. So this window shows an overlap, allows us to overlap the experimental images with the Allen coordinate space. And on the right is the Allen coordinate space in sagittal view. The line through this is showing us the atlas, where we are in the atlas. So we can move this line. Let me back up a little bit. So the window here is for section registration. The top view is we use this for determining, or for when we actually go through and do the registration. The adjustment window allows us to move within the Allen coordinate space plane. So you can see I can shift, and you can watch how this red line that is cutting through at this level of the in the z-axis and showing the image, the Allen image on the left. I can go to the back of the brain, the cerebellum, into the midbrain, into the thalamus, to the front of the brain. So this is how we can navigate through this space. So we can also, this overlays the experimental image with that Allen image space. And we can, that's done with the visualization. We can change the color so that we can actually make them two different colors. And then I can change the intensity. So now this is showing in red the experimental section and where we are. And so now I can move through where I am in the experimental section using the ortho view. So now you can see this is now navigating through the experimental sections. So what we're looking for is to try to match, as Nate described, one of the, not problems, but the strategy behind registering a brain that's been reconstructed in this way to the Allen coordinate system is that the sections that we cut are not perfectly orthogonal to the 2-photon block-based imaging that was used to generate the Allen space. And there's compression in terms of the in the y-axis, and that can be adjusted both by doing an atlas calibration, but can also be adjusted when we do the registration here. The other thing is that when we look at the in experimental brains, the position and shape of different structures gives us an indication of where we are in the rostral-caudal axis. So as anyone who's gone through these brains knows, the shape of the hippocampus is very telling about where we're located in the rostral-caudal axis. So in the front part of the brain, in the rostral sections of the brain, the hippocampus sits right above the thalamus and has this particular shape. As we go caudaling, the hippocampus grows larger, it starts to wrap around the thalamus, and in the midbrain, it wraps all the way around the midbrain. So the shape of the hippocampus then is an indicator of where we are in the rostral caudally in the brain. Similarly, there are other fiber tracts that give us a similar indication, like the optic tracts as they come into the brain, where they're located gives us an indication of where we are. The mammillothalamic tracts, the shape of structures like the amygdala, the basolateral amygdala. Rostrally, it just starts, and then we can see, and we can estimate where we are located in the Z dimension based on the location of these structures. So the strategy that what we use for registration to the Allen space is we want to take this section and find its best fit in the Allen coordinate, in the Allen space. And so you can do that by getting to a space, getting to a position in the Allen that's fairly close . I'm doing this by using the scroll bars for the shifting. You can see again where the this plane through the Allen space viewed here. You can see how that shifts in the z-axis. So I want to find a place that's like close to where we are in my experimental brain is close. And so I can see that the hippocampus is about the right size and all of that. When we get to this point, then individual brains vary in terms of the XY scaling. That's just happens. So we can move the brain itself. Now we're moving the Allen space relative to where the experimental brain is. We can move in the y-axis. We can move in the x-axis. So we want to line this up as well as we can. And then sometimes do some minor adjustment in the scaling. That's pretty good. So what I can see is that the hippocampus at this level might be in the comparable position. So that means that the hippocampus here in this plane is accurate. However, if you look at where is an example, the shape of the amygdala or the location of the optic tracts coming into the brain and the mammillothalamic tracts, you can see that these are not in the same plane as in the orthogonal view of the Allen space. So what we were trying to do, the strategy behind this is what we want to do is to find a plane within the Allen space that best fits our experimental section into that Allen space. And so we can do that by rotation. So these are the parameters that allow us to move within the Allen space. Shifting, these are just straightforward, shifting in the XY positions and Z. And then these are rotations. So this allows us to, move So what this is doing is it's cutting through, and it's the hippocampus at this level, and the optic fiber tracts are at this level. To get that space within the Allen, to find that space within the Allen, we have to make this shift the x-axis or rotate in the x-axis. Also, there might be some difference in the asymmetry between the right side of the brain and the left side of the brain. And so you can see that I can shift in the rotate in the right and left. And so this red box that Nate pointed out before gives us, visualizes the offsets in the x and y axes. And what we want to do is to find the closest that we can to the Allen space. Now looks like I've over rotated in the X and probably a little over rotated in the Y. And also I need to shift back a little to make the hippocampus the right size. Now going back, the optic tracts are fairly close. So this is just to visualize how the plane through the Allen space, what we're trying to do is to find a plane that best matches the experimental section itself. So we can go through and manually do this. However, the more straightforward way to do this is we can do this, the program itself does this. And so we can clear this, start over. Want to give it a little bit of a head start by getting to the right close. I get close by just getting, close where the hippocampus is. And now if I run linear, hit the run linear function. Now the program itself is going through, and Brian, you might describe what the program's doing since you wrote this part.
Yeah, sure. So yeah, so I think that's great the way you went over all of those parameters. The way I like to think about this is that red plane basically represents the plane that the knife cut through the brain while you were sectioning. And we have all those parameters that model how that knife sliced through the brain during the sectioning process. And then we also have in the registration engine, we have this way of adjusting those parameters automatically to find the best match, for image content between that experimental brain and the image in the Allen Alice space. And that's what that automatic registration does. And you can see the results of that now.
Yeah. So what we're
hey, I have a
yeah,
quick.
so what we're doing is we're trying to find the best fit here for this one. And then the idea is that if we can find this same offset should be able to be applied throughout the whole brain. And so the next part of what we do is we see, okay, does this offset, can we apply that to other regions of the, other levels of the brain? So we'll go rostrally, maybe that's a little too rostral. And when we do this, we can see that it's offset in the it's offset in the y-axis, which is okay, because it's going to fix that later. And now if we go back and forth, we can see where we look as, is the anterior commissure matched up correctly? And it, for the most part, is. The anterior Nate, did you have something to say?
I just had a minor bookkeeping thing. We've got a lot of awesome questions coming in. I just wanted to acknowledge that to people. We will at the end try to get to as many of them as we can. There's a question panel over on the right in the interface itself of GoToWebinar. Feel free to keep them coming. They're really getting some great questions. But I just wanted to make that note that we will, and if we don't get to them in this session, we get to everything via email. So yeah, I just wanted to acknowledge that quick.
Okay, so what I'm doing now is I'm going to different levels of the brain to see how well this offset that we established for section 55, which was through the hippocampus and thalamus, if that same offsets can be applied to other parts of the brain. And in different levels, we use different markers. So the anterior commissure here is a marker that is a fiber tract that its location gives us an indication of where we are in the z-axis, and so that's a good one to look at. And so it does look like that now it's fairly well lined up. And then I'll just do a few more.
And I'll just jump in to say, of course, that the model that we have of the brain being sliced during sectioning. When you're sectioning, you keep all the planes parallel. The knife always travels through the brain in parallel planes, and that's why this particular approach seems to work really well.
Yeah. So this is another. When we're looking at the fiber, the fimbria, looking at the fiber tracts there, you can see how well that matches up. So by going through this, I can tell that this offset, these angles, and as Brian just said, the strategy behind this is that the offset, the knife, the relationship of the knife to our brain when we cut it is constant all the way through cutting. At least it should be. And so if we can find what that offset is, this is basically an exercise to find what the offset is from our, the cutting angle to the Allen space. And after we do this, you can say, okay, then we can go back to the original section, which was section 55. And there's typically when we cut these brains, it's about usually about 120 sections from the frontal pole through to the medulla. So now we're back to section 55. So what we've done is that this offset has basically applied or changed the parameters of the plane cut through the Allen space. And so, and this I'm just repeating, those parameters are rotations in the X and Y dimensions and Z, but we didn't deal too much with Z, and also shifting the section in the X, Y, and Z planes. So these are linear transforms, and by that we mean that the same the parameters have been altered, but they've for each section, but they've been applied across the entire section. However, in these sections, there, as Nate pointed out before, there's some each section generally has some deformations where there's not perfect alignment, and that can be due to, like when there's injection sites, there's depression of the cortex, or there might be minor tears, or the tissue's not perfectly flattened, or things like that. And to make those adjustments, then we can run a nonlinear transform. And so the example I wanted you to look at is right here, is between, this is the experimental section, and this is the Allen space section there. And you can see that they're not perfectly aligned, not perfectly matched up or lined up. So if we run a nonlinear on this, it will now apply local transforms and fix that. And again, Brian, I think you're best describing this.
Sure, I'll jump in. So yeah, as Chip said, the linear registration is really good at finding where in the brain we are, but it doesn't represent all the variation within each section. And so that's what nonlinear registration does. Instead of the handful of parameters that we have to represent how a knife cuts through the brain, the nonlinear model has hundreds or potentially thousands of parameters. And what those parameters enable us to do is to incorporate local adjustments that account for tissue distortion. And that can be distortion that's caused by the cutting process, by the knife that cuts through, or it can also just be differences between animals. And so that's what the nonlinear registration does. And so with linear registration, we're mapping a plane onto a plane, and straight lines stay straight. With nonlinear registration, the straight lines don't stay straight anymore. And so we can have local deformations that better match the structure that we find in the brain. And Chip, when this finishes, what I'd like to ask is if you could take a look at some of the regions and show how we can draw contours around the regions in the brain, and look at that anatomic structures that are mapped in the Allen atlas.
Okay, watch. All of a sudden, it's about to finish. Okay, so you saw when it finished, the edge of the cortex, it snapped together. So this is what the effect of the nonlinear registration is. And thanks, Brian, for reminding me what I always forget. Anyway, so the process that we use then is, after we've identified this, the optimal transforms, the parameters that are required to match that. And again, the parameters that are used for the linear transform are, shifting in the X, Y, and Z axes, and rotating in X and Y, and all. And so then we can go and lock those, and do multiple. So we could do the whole brain, which would go from the front of the brain through all the way through 120 sections. That would take probably, it takes usually between an hour to an hour and a half, sometimes depending. It's completely dependent on how fast your computer is, but it takes a while. So rather than do that, I'm just going to start with section 55 and just do a few of them. 58. And now what the program does is it goes and it starts, since section 55 has already been registered, then it's going to move on to section 56 and apply the linear transforms that were used for section 55 to section 56. And then after it's finished with that, it's going to move to section 57, 58, and go all the way through the, and it would, if I had set all, it would have gone all the way through the brain. You can see that this angle, but the point to notice is that these parameters, that the angles used for this, the angle of the slice is maintained for each of those sections. One of the things, the major thing that it does is that it does adjustments in the y-axis, because when we generate this whole brain volume, one of the things that's difficult is to get the y-axis to be, or to get the sections aligned perfectly in the y-axis. You can see in the normal brain, there's some curvature to that. With BrainMaker, sometimes it flattens that out. So this is one of the things that it does, and it does anyway. So it's gone through now, and it's applied the linear transforms, in this case, just the 3 sections. And then after it does those 3 sections, then I would, and now run the nonlinear part. And it does the same thing, and it's going to go through, and what we showed before for the individual section, it's going to apply the nonlinear transform section by section all the way through. And again, this takes about an hour and a half or so to run. That's all just, once you set it up, then you can go leave and do other things. So it's just computing time. It's not actually, you don't actually have to do anything. It's doing it automatically.
Chip, toward that, can I ask, like what, I guess per day or per week, what is your workflow in your particular lab for how many brains you can get through at this resolution and this rostral caudal extent?
Well, going through this part, as I just said, the major time is the, well, we'll go through when the cell detection can take an hour to 2 hours. Again, it's automatic. We'll do we'll do a webinar on that. Sectioning or doing this registration, it takes about, I'd say, between 2 and 3 hours of computing time. It takes, 10 to 15 minutes to set it up, and 2 to 3 hours to actually have the computer run the program. So, we can run 2 to 3, on average 2 brains a day on using this program. A major limiting factor in all of this has to do with the imaging time. So the imaging time really is what's rate limiting, because we imaged, and so that's again another webinar that we'll go through and describe that. On average, we have 2 microscopes that we run pretty much all the time, and we process, myself and Ron Piletsky, who works with me, the 2 of us process about 4 to 5 brains a week, or can process about 4 to 5 brains a week. And doing a lot of other things at the same, because a lot of that's just the computing time that it takes. So when this is done, I'm going to Brian, you're supposed to remind me now.
Yeah, I really want to take a look at the anatomic regions in the atlas. Yeah.
Okay.
I think this is
so
cool.
We can do this in. So what I can do now that they've been registered, oops, it's still registering.
I think it's just the last section is finishing up.
So what Brian's referring to is this program does enable you to pull up the different.
Oh, sorry, there was one more section to get.
Okay.
So what you're looking at right now, Chip, that looks like, so that's those are all the anatomic regions that are annotated in the Allen Atlas. So the Allen Atlas has both reference data and also anatomic region delineations. And this is what you're looking at now is the hierarchical view of all the anatomical regions that are annotated in the Allen Atlas.
Right, and so what we can do is that we can actually then go and click visualize these, and we'll provide an overlay into the atlas itself. I mean, into the registration itself.
Yeah, and you can see as you're moving your mouse, you can see that in yellow below the image, the overlay image, you're seeing the anatomic region, the name of the anatomic region that your mouse is hovering over.
So we're in the lateral habenula, the dentate gyrus, CA1, CA3. Does it get CA2? Yeah, even CA2. That's pretty impressive.
Yeah.
Amylothalamic tracts, basolateral amygdala, Now it's done, maybe. So
Yep.
if
I
I
think now it's done, yeah.
so if I click on, do I have to click on this?
It's control click. If you control click.
I use this program all the time, and yet there's still things I don't know about it. So this is showing the medial dorsal nucleus. And let me just. So anyway, this is a way that we can go and visualize the different.
Yeah, and I don't know if it's clear, but as you're clicking on those regions, we're also taking you to where those regions are in the hierarchical view, in that tree view, and so you can see where that structure is in the whole hierarchy of all the anatomic regions that have been delineated.
And then you can see it's shown also in 3D.
Yeah, we have a model of every single one of those structures in 3D that we can overlay in the 3D view of the atlas.
You could then save these structures. I'll just throw in a bit here and go to some of our other software analysis suites like Stereo Investigator for different types of stereologic measurements, for instance, too.
So after it's done all of that, then when we hit Map Data to Atlas, it takes all of the markers that we've identified that have been marked in this brain, and it transforms them into the coordinates in the Allen space. I'm not going to do that now because it takes a while to do that. I'm going to actually turn off section registration. Anything else I should? Oh, too late. And now I'm going to close the. So this is the image file that's been generated that's registered to the Allen space, and now this is again, we can navigate through the brain, and now it's showing all of the sections through the whole brain, but we want to see them only through the section that is, in an individual section in the Allen space. And so here you can see the cells that are marked the injection site, and then the marked cells in the layer 5 of the cortex. And each of these gray regions is one of the regions that's been marked in the Allen space, and it has a number of cells that have been marked. That's what's generated and put into that into that Excel sheet. And now you can see the fiducial markers and see how well they line up with the fiber tracks around the globus pallidus. We use these fiducial markers to determine how accurate our registration was. And so this is showing the, you can see how the fiducial markers are fairly well lined up with the original, these had been marked in the original images, and then we want to see how well they line up with the, in the Allen coordinate space. So what we
here
find
you switched over to that 2-photon image as opposed to the annotation volume.
Yes, so this is the 2-photon image, not the so we can view in the atlases is displayed in 3 different modalities. One is in where the structures have each structure has a unique grayscale value, or can be colorized, or the original 2-photon images. Anyway. So the Excels, what we can do, as I said, with the Excel spreadsheets, the data that's, let me just do one more thing. So the data that's is presented in the spreadsheet that has the cells per, in each structure, then that is, that allows us to then do things like, in this case, we could compare the number of neurons in primary motor cortex or secondary motor cortex between the different experimental conditions, the normal Parkinson's model, Parkinson's with L-DOPA treatment, things like that. And at that, I think I am about done. Turn it back to you, Nate.
Very cool.
That's awesome.
Yeah, that's really great, Chip. Hey, one question I was going to ask. We're getting near the end here, but we do have time for a couple questions. I've been going through them, seeing what the most relevant ones are. But I know that we have a number, a rough number, or actually, Brian, maybe it's not rough. You can talk to this on the accuracy of this. I know we've talked about it in numbers of neurons. Do either of you have anything to speak toward that?
You're talking about the accuracy of the registration. And so we've actually done this in a publication with Mac Hooks, and we went through this. And what we determined was that the accuracy of the registration was within, on average about 80 microns, which when you think about it, that's pretty accurate. And the way we did that was to take these fiducial markers in, a number of different regions of the brain, and and determine what the offset was between what the registration did and what the actual position was in the Allen space. And so on average, it was about 80 microns.
Cool.
Accuracy.
Yeah, that's I'm going to make myself the presenter now and just throw up a couple slides and do some bookkeeping and hopefully take a couple questions here in the last few minutes. All right. So just to acknowledge a couple things, these are folks who've worked on this. This is the funding, and of course, I really want to thank you, Chip and Brian for this. Is really powerful stuff, and really always fun to chat with you guys about this, and hopefully that was useful for folks. The other thing I'll mention is I'll remind folks that this is also an ongoing series of webinars. And next, Chip, what do you think we're going to talk about? Maybe cell detection or brain maker, one of those?
Actually, we wouldn't mind getting feedback from people of what they'd like to see next. So the things we need to talk about is cell detection, how to generate the whole brain volume, and then other lingering questions from the registration. Any of those things are open. So we use these programs all the time, so it's really dependent on what people feel that they would like to hear next. I think we might.
I see a lot of great questions in the question list too, so we should try to get to some of those as well.
Yeah. The other thing I'd mention is that the Chip, that's a fantastic point. Is we want that feedback, so definitely fill out the survey after the webinar. That would be super helpful to us if you have the time. That'd be great. I'll just mention that every Friday is TGIF Friday with our amazing technician TJ in our MDF Labs, which is our CRO. And he picks a part of our software and does a webinar on that every Friday. And it's real hands-on stuff, so you get to see how this stuff actually works. And then we're also on Tuesday, February 23rd at 2 p.m. Having a webinar about data standardization and fair standards in NeuroLucida, so that you can annotate microscopy image data with these online and well-established curated vocabularies. So that'll be a really cool topic right now. With that, let me pick a couple of questions here, and I'll field them to all of us. I've got one here. We have one about how do we register the mutant brain but still maintain the genetic distortion like the P10 null brain?
Can you repeat that?
Yep. So someone's asking basically, if we have a mutant that has significant distortion, how do we accommodate for that?
So I think that I can try to tackle that one. It would depend, in my mind, I think it would depend a little bit on what type of distortions are present.
Right.
So I'm not familiar with that type of mutant in any way, but.
So that's when we can dig in a little more on.
We might want to actually, consider doing like a pathological, like a webinar on pathologies. I think that would be really interesting. So the nonlinear registration that we do can sometimes address those issues, and I guess that's what I would say. Is we find that that's sometimes sufficient.
The other I have a lot of questions. That's why I'm going through them. Yes, a trial is available. You can go to our website for that under products and request a trial. The other thing that you can do is, let's see, free trial. will attendees automatically be advised of upcoming webinars? Yep, we definitely. If you're here and you've not opted out, you will get an email from us. A lot of questions about modality, whether it's NISL, NUN, DAPI, and I answered that partially in text, but do either of you want to talk to that?
Brian and I, we talked about it a little bit already. So originally, you know, the modality that's used in the Allen coordinate space is the 2-photon image and all. And so what we found, and what we've actually found is that we can use DAPI. Actually, I don't use DAPI, but fluorescent NISL, DAPI, other modalities actually do work fairly well with the 2-photon atlas that's used. You'd have to go through and you know, and just try them. So actually, it's fairly flexible in terms of the modalities that can be used for that 2-photon, which is somewhat surprising because the you know, the image, the brightness, for instance, of the cortex is very different, and the difference between white matter and gray matter in the 2-photon is very different than what we see in NISL, and yet you know, the program that Brian's written actually does a pretty good job of accommodating those differences. So even though we use this average NISL brain, there's, you know, quite a bit of flexibility that can be used.
Yeah, and the only thing that I would add to that is just to say that the labeling, whatever labeling you use, it just has to show the anatomic regions in some way, right? It has, I believe it's the cytoarchitectural stains are the ones that we want to look at. Because what we're looking for in the registration is some correspondence to be able to recognize that there are similar looking regions. They don't have to appear exactly the same. They just have to be labeled in some way where we can say that's, you know, that's the same region.
Yeah, so any stains that provide some contrast between different structures is basically what Brian's saying, and that and so that we find that there's a wide latitude that can be used.
Right. That's awesome. We're 2 minutes over. I think we've done really good on time though. This was super excellent. The other thing I'd say is we have a really cool discussion area. We have forums that you can go to. You can find them on our website, mbfbioscience.com, and under resources, you'll see a link to forums. There's also a link there to webinars. So that's mbfbioscience.com under resources. And feel free to engage us there. And you'll be getting emails about subsequent webinars if you've signed up to get them. And with that, thanks everybody.
Thanks
Thank you.
everyone.
Goodbye.